Okay. Do you guys see slides?
Yes.
Okay. Um, okay. So I pulled some of these together um, about this rare disease brain workshop, which was phenomenal. Uh, I I I could go off on so many tangents and tell you so many different stories about why this was so good and so important for me to be at. Um, but I'll just give some context. This was hosted by the Rare Epilepsy Network, of which DESSH is a member. It was in partnership with Global Genes and with Mazi Therapeutics. It was meant to be a true workshop where there's actual built-in activities and sessions meant to help you explore your research questions and give you a real overview and like full picture of the drug development landscape. And I felt extremely overwhelmed while I was there because it was literally 7:45 in the morning until after dinner every 3 days in a row. And it was super intensive and filled with people just like us being presented to by people who were just like us 10 years ago. And it really was exciting to see the progress that patient groups were able to make. And then we were in the room with stakeholders all across this industry who will meet and need to interact with each of them in the process towards something that we can actually deliver to patients one day. So I invited, Lucinda was unable to attend because of her daughter had surgery. So we invited last minute Alyssa Bierstead, who formerly Alyssa Erickson of Harrison Gables Lab. She's our grad student there who's been working on the WAC mouse models. She's presented at all of our clinics, so many of you know her. But I was so grateful that she was able to come. And honestly, we would not be, I would not be, half as, um, I wouldn't have been able to function there without her, honestly. Stuff was pretty over my head, and she really helped explain things to me in a way that I could understand and hopefully translate to all of you. People at the workshop were also very impressed with her. We had a couple of office hour meetings individually with other CAG leaders, also with biotech leaders who were asking, deep science questions, and she rolled with it, no problem, to the point where they were commending her on her knowledge. So super excited that she was there with us. So what I want to go through with you is I tried to really, for the board's sake, take a super high level 30,000 foot view of everything and hopefully give enough information that we can end up leaving here with sort of an idea of how we can move forward towards treatments because that's honestly what this is all about. Um, so I just started. I'll start with some topics of what I learned, how it applies to DESH, and then what I think we should do with that knowledge. So, some things that may be apparent to many of us that seem apparent on the surface came to light for me, I guess, in a new way at this workshop. So, not all genes are created specifically or created equally. So, gene expression is specific. That means that I was always confused about this. Every single cell has the same DNA in it, right? Like every cell in all of us still has the WAC gene in it. But certain genes only work in certain cells, only do something, only function at certain times in your life, and only make things happen in certain areas of your body. So, where a liver cell would activate, there would be a different set of genes activating things in the liver cells. WAC is different, and I'll tell you how later. But for now, know that there are different types of genes and different types of cells that do different types of things all over the body. Um, Another separation, or I guess idea that I walked away with, is that not all mutations of genes are created equally. So there's different types of mutations, which we've all heard of now. We know that there are point mutations, deletions, insertions, duplications, and that those do certain things to your cells. How they make your cells behave could either not express a protein correctly, could overexpress a protein correctly. There could be neutral cells. So the way that mutations affect your cells are different across all of your genes. So this rare epilepsy, like I said, was the host of this, and global genes, which is a rare disease. So you put those two things together and you'll notice that this was really geared towards all neurodevelopmental diseases. And so those are really defined as mutations that cause atypical development of the brain. It's not just a brain issue. It's not something that, you can, a brain disorder in the sense that it, it affects only the brain, or that it was caused by some type of degeneration later in life as you age, or some environmental factor like a brain injury. It really is defined as starting at development, things aren't working correctly. So you can imagine that many of those causes are from genetic mutations. And we all know the manifestations of these neurodevelopmental disorders. We see them in our children. And then what we learned about, so what do we do with this group of symptoms that we classify as neurodevelopmental disorders? Well, there's a lot of different treatments for genetics that we've all heard of, but I learned a little bit more about. And so samplings of those are repurposing an already approved drug, finding a new drug, small molecule that would work, antisense oligonucleotide delivery, gene replacement therapies, and gene editing are the most common, I guess, really 5 top head honchos of ways to treat genetic syndromes. So where is whack in all of this? Like I said, not all genes are created equally. So I said before that different genes do different things, and some genes are only work in certain areas of the body. Well, whack is expressed ubiquitously, which we've all heard. But what that really means is not only is the gene in every cell in our body, like every gene is, because every DNA is in every cell, but it actually is working in every cell in our body. It has an underlying function that is expressed throughout our lifetimes and in every cell of our body. So we've heard this before, but I guess it landed for me that treatment now, actionable treatment on that, is going to be a lot different than something like sickle cell disease, which is targeted to bone marrow, or a lot of liver diseases where replacement therapy is really just changing the gene within one type of liver cell. That's not true for DESSH. We're everywhere and we affect every system. We're in every tissue, we affect every system. And so, and it's not just at one point in development that we know of yet, that quack is working. It seems to really be the source of life. It's a functional, one working copy needs to be there at least to sustain life in an organism. So going into, not all mutations are created equally. So the type of mutation, or the thing that's wrong or missing in your gene, everyone has 2 genes of the same kind. The change in them can do different things. Fortunately, no matter how the change occurs, meaning if it was a deletion, a point mutation, or a duplication, it still seems to have the same effect. And what is the general consensus is that it is a haploinsufficiency. We've heard that word in all the literature. What does that mean? It just means that it's loss of function. It just means that there's 2 copies. One of them's fine. The other one is not working properly. And then the sort of hypothesis of that not working properly is then, can we if that were to work properly, would everything be restored in the symptoms that we see in the patients? I think that covers this entire thing. Okay. Um, so as we know, it's a neurodevelopmental disorder, and we see it in these manifestations of our kids. And I want to point out that everyone there seemed to have the same manifestations. But I think it's important here. We're all a neurodevelopmental disorder, but there are some nuances within this space. As you can imagine, there are hundreds of rare diseases that are considered neurodevelopmental. 30 patient groups were, or I guess 20 patient groups were really covered at this workshop, or had attendees at the workshop. But something that I learned. So there's a lot going on in neurodevelopmental around seizures and epilepsy. As you can imagine, it's a huge factor. It affects quality of life. It has a big economic burden. And so a lot of attention was paid to DEEs, or developmental epileptic encephalopathies. DESSH is not one of these. DESSH has seizure, but what the DEE designation means is that seizures are usually not controlled by medication, and they sort of have this effect where the seizure is causing developmental delay, and the gene is causing a developmental. So it's not just the way that a gene works that causes a developmental delay in other syndromes. Sometimes seizures compact that issue. Because our seizures, first of all, not everyone has them, and second of all, they are pretty responsive, well responding to medication. We don't really fit that designation, and in a sense, that's a nice thing. That's a clue about our syndrome, in the sense that we now know that our developmental delays come purely from the gene. They're not being affected or caused by another symptom that the gene does. So it gives us some understanding that DESH does something specific with development and intellect and cognition. I'll also point out another thing that was different among us is our neurodevelopmental disorders is everyone does present with some form of seizure, some sort of autistic feature. Everyone has a developmental delay, some type of intellectual disability. And just like in DESH, there's a range from mild to moderate to severe in a lot of these. And one topic that came up as another side note with a great PAG leader, Mike Graglia, he really, in our office hours, talked about the need for some groups, I guess some more, maybe even an umbrella organization that really covers the phenotype of severe aggressive behaviors and children or adults who end up needing to be moved to residential home for safety purposes, or who become wards of state because they're unmanageable and their behaviors are so severe. So that's a conversation that I picked up on because I know that's something that our families are really dealing with, and that I fear for in my own family. So we're picking up that conversation with a couple other PAGs that have that same phenotype, so that we can see how we might be able to work together. Okay. So I talked to you about all the modalities of treatment before, but this really gives you a little piece of what the modalities are, and if the likelihood that they are a good candidate to treat DESH. So small molecule drug repurposing are just drugs, medications. A lot of times they help with symptom management. And they really do well when you're targeting something specific in a gene, when the gene needs to turn an on switch or turn an off switch to make something else happen. It's a very simple, direct measure. Small molecules work really well for that. But as we know, WAC is pretty pervasive, and it would not necessarily respond well to something like this. And so that's something to know, not that it's not an avenue we can't try. Certainly there has been clonazepam as a drug candidate in this category. But if we really want to get down to treating the underlying condition, you would move into something like an ASO. ASO is what you think of. It's not a virus, but it's what you think of when you think of a virus and an RNA therapy. It's an injectable, and it's a nucleotide that's basically resequenced so that when it attaches to a gene, it ends up, you know, sending a new message to the proteins on what to do. It's difficult in the sense that it needs to be given in terms of a treatment for patients, as the delivery method is usually through a spinal tap, because you could, I guess, inject it directly into the brain, which is done, but that's incredibly invasive. So a lot of times it's injected right through a spinal tap into your central nervous system. It can't be, given like a regular injection because it doesn't really pass the blood-brain barrier very well. So you get it into the central nervous system. Part of the problem with it is it can degrade. So you're looking at multiple treatments for that, but it is a direct hit on the gene and what it's doing. And there's been a lot of success with these in reality, and DESSH is a good candidate for ASOs. And sidebar, I just got word from Harrison that part of this NIH grant funding that he has is in partnership with another lab, and he threw WAC into the mix. And one of the ideas is actually getting developing an ASO, or seeing what kind of ASO work they can accomplish at their lab for WAC specifically. So that's like super progress. The next sort of mode of treatment would be gene replacement therapy. And that basically is just throwing in a new gene. So it more or less replaces the bad gene. And it can be done for any mutation type. So it's really good at, it can cover, all of us. It's not necessary, it's precision therapy, but it's a broader precision in the sense that no matter how your copy is mutated, this would fix the problems. The only issue comes down to we don't know how much would need to be dosed into this replaced copy to make functioning of our cells work. It could lead to potentially overexpressing WAC, and then that could cause other downstream problems. So you need a lot of validation. Bottom line with all of these, you need a lot of validation before you can go into, down the pathway of using this in humans to treat humans. But I will say another positive, gene replacement therapy doesn't work for a lot of genes. And I'll tell you why. It's the delivery method. Different genes are different sizes, something else I didn't know. And so the way that there's delivery to these genes is through these vectors and capsids, where they would, I guess, put in the substance and the new gene, the gene replacement. And if your gene is too long or too large, they have a really low payload. These are carry load, the vectors that deliver the medicine to your body. So DESSH actually is small on the scale, or WAC is small in comparison to many genes, and actually would be fine to be transmitted to a human in this way. Then we get to gene editing. This is where you hear of CRISPR all the time. And this is where you're going in and you're fixing the actual mutated copy of your gene. This is extreme precision therapy, almost on an individualized basis. Unless someone has the same exact mutation, you are developing a therapy for each individual person with DESSH. Right now, things are too early for gene editing to work in any neurodevelopmental syndrome because there is no delivery to the central nervous system at this time. that just hasn't been invented yet. So they've invented CRISPR and the editing mechanism, but now they really need to invent delivery into the central nervous system. Um, and then the big ethical question of this, of course, is it can't be reversed. It can't be undone. With an antisense oligo, you will be able, it degrades over time. With a small molecule drug, if it's not working or causing damaging effects, you can stop it. With gene editing, that's just not possible. And so you have to have, I mean, layer upon layer upon layer of validating evidence for anyone to even touch that as an approval. But we don't have to worry about that until someone invents a way for it to be delivered anyway. There was a fifth modality that was touched on, tRNA suppression, and that really would only work for certain mutations within DESSH. So not all of our children have the same type of mutation. For example, Leo is frame shift. His frame shift mutation would not work that. It produces a stop codon, and everything after that is kind of done. tRNA suppression was really easy to think about it when they explained it like a find and replace in a Word document. So if you have ACTG patterns and you want to fix all your, and it's switched so that an A is where a T is supposed to be, you do a find and replace, and you just for a certain section of that gene strand, and you insert it where it is. But it can be finicky in the sense that, maybe there is an A where you want there to be an A. And if you just find and replace all the A's to T's, you're causing harm. So it can be good. And actually, Alyssa is following up with someone who works on tRNA therapies to pursue this more for those with nonsense mutations of DESSH. Okay. So moving on, I talked about a lot of therapies and treatments, but how do these treatments actually get made and given to us? It's an entire ecosystem of people, and really at the center of it and connecting a lot of us, actually driving it, as you can imagine, are the patient groups. There really is no need for them to be doing any of this without patients. There also really is no I guess, awareness that it needs to be done without patients. And so our role, and I'll get to this soon, is really about getting these folks together. But first we need to understand what they all do and how they all relate to one another, which I got some sense of. So academia, as you know, we have our Michigan State mouse model with Dr. Vogt. We have our Gable lab at WashU. We have even Dr. Kim in Korea is attached to a university. And so these are all places where basic research happens. And basic researches are like what we've been in the past 5 years, are just developing all the tools that science really needs to start being able to test therapies. And a lot of that comes from academia. When you start moving into treatments and modalities, that's where you get biotech and their involvement. You sort of need a strong business case and a strong scientific case to get biotech interested in your therapy. And once they will end up taking over a lot of the treatment experiments that need to happen. They'll also converse a lot with academia. And so there's some back and forth and sort of overlap between what they each do in terms of finding the underlying conditions, validating certain hypotheses, and then developing therapies out of the biotech. Also, once you get to the biotech stage and you have sort of a viable therapy, you want to get the FDA involved pretty early if you want to bring this as an actual path to treatment. And that is because FDA really needs to, they can be involved from the beginning. I used to think that science was science, and it actually is more of an art form. So things can be tweaked and changed in terms of what biomarker you're going to study or what change you're going to go after. And FDA can inform a lot of that in advance for you to know what they would accept as evidence for approval versus not. And so conversations go back and forth. changing of your natural history study. I thought natural history studies like needed to be set in stone and there's a certain way to do them and we need to follow the protocols. And I haven't been able to really nail down what that is. And the truth is, no. Yes, there's a version of a clinical trial natural history, but you won't know what that thing is until you've gotten to the point where you're finding treatments and talking to FDA, because they're going to trim down and narrow down all the stuff that doesn't matter in your natural history study and hone in on things. And you know what? While you're still validating stuff and the DESSH clinic is still going on, we might add something to the natural history that we haven't collected before, or find other health records from patients that can add to the natural history. So everything is kind of evolving in a way that I didn't realize before. From FDA, you're obviously after approval, not even after, but during this FDA approval, you also need a way to get it to patients. So this is where drug repurposing becomes a really hard business case, because while you don't need FDA approval, you are going to need someone that's willing to deliver it to patients, and then find a payer or an insurance company who's going to be willing to cover it for a non-indication. So you have to pull them into the mix to make sure that once you develop. So Amber Freed was there and she developed a gene replacement therapy for her son. And he basically went through FDA approvals as an N of one and she got him treated. But they don't have a way to treat this for the rest of her community at this point. They need to try to get this to scale. And that's where they're trying to entice pharma. And then how all of this happens, money. Money, 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 and how we engage with really large, not even donors, but super investors, like multi-million dollar grant investors, life science, venture capitals, folks were there. We spoke. They talked to us at length about what they look for and how they're going to be able to tell their investors that this is worthwhile. And you know what? The one thing that they did say is, while biotech is normally in the room with them, it's the patient stories that get them, motivated and moving, even though at the end of the day, they'll all admit it's a bottom line numbers question. But moving them to really understand the hardship helps them at least be able to show their stockholders why they're doing the work that they're doing. So like I said, all these people are talking to each other and they're all centered more or less around a disease area because of us. So if I take one big insight from this entire thing, from everything I shared, from the whole 30,000-foot view, it's that our foundation must be the authoritative source on DESSH. A lot of times we've all experienced it as parents. We go on the DESSH chat, we say something, someone has an idea. Well, why don't we check with Dr. Shinawi on this? Why don't we see what the researchers have to say about this? Yeah, they have some fantastic insights. They're driving so much of what we know. But at the end of the day, they're also beholden to their own, they have to worry about being a liability in any type of treatment that they might recommend or suggest. They will oftentimes not truly not know the answer because they haven't experienced it or seen it in another patient. But guess what? We have access to a bajillion patients, and there's a lot of data that we can be collecting even outside of registries that would help us navigate and help, not just help parents answer questions, but really be the catch-all for anyone that needs to know anything about DESSH. So we really need to understand the science. This is something, I couldn't have an intelligent conversation about DESSH before I sat with Alyssa, before I talked to some of these other folks. And I still don't even feel a quarter of the way there when I'm talking to folks in this industry and in science. But it like I need to go back to school for this. Like, we have to understand everything about this. We don't have to become the scientists, but we need to know how to talk about it and what it all means. And we obviously, all the things, ask better questions, challenge every researcher that brings us something. They talked a lot about how some scientists will sort of like, they'll geek out over like a cell line and say, "Ooh, I saw this weird thing happen. You think we should chase that down or track that down?" And sort of like, I needs to stay on the prize, and that's sort of what we're here for. We need to keep everything moving forward. We're like the wheel, the engine that keeps this bus moving. So, how do we do that? I think I have a couple of priorities for us. The first one is the infrastructure side, which I feel like we've been doing for the past 5 years, just building natural history registries, finding new patients. For the past 5 years, our whole goal has been finding and connecting patients. For the next 5 years, our goal gets to be finding treatments for these patients. So we still have to continue doing that work that we're doing. So growing the database even larger, expanding all of our data collection efforts, like I mentioned, um, strengthening global DESH infrastructure. And this does not need to be through like legalese and contracts. This can just be having a banner holder in every country that says, "I'm the DESH go-to for this population." And they get a DESH India page and a DESH France page as our part of our website. It really helps. There was a case study that talked about a guy who, our numbers are so small, and they really wanted to see if they could really leverage the full patient population. And some of these biotech people are like traveling to other countries to meet other families with the same syndrome. And they would have one experience where there was like a well-formed group community, could answer their questions. They could really get a sense of things. And then they would show up at other places, and there would be no information for them to find, and no patient, organization, even though they were all part of the same sort of syndrome. So having really strong people on the ground locally in these places, we just need the same logos. They need to be part of us in that way so that we're unified, but really still have local efforts on the ground in all these places. And then really develop our DESH scientific and business case. And this can be a simple pitch deck that I can attach every time we talk to a new person in industry. Just all about DESH, what we know about it, what we don't know about it, but with a slant towards scientific opportunity. So building infrastructure is necessary, but it does not create the treatments. We also need to have some real research priorities. So it felt easy before, where we just knew we sort of had to build up this toolbox of things to get to this point. And now I feel like we're at this point. So we need to start deciding how we want to attack and use these tools to develop something that will help us, help our kids. So what's the hypothesis for DESH? And I would propose that if it is fundamentally a disease of insufficient WAC expression, it's loss of function. So my hypothesis here, I'll get to on the next page, but I really want to clearly define what that is. I want to evaluate whether re-expressing it actually fixes the problems that are associated with DESH in people. And then I want to see what modalities would attack sort of DESH patients that we have right now by mapping, modalities to our specific variants in our population, and then identifying paths to proof of concept. How are we going to get there? So my last slide really is my proposed strategic question for all of us. And if this is a question that we can rally behind, it'll really sort of guide our next 5 years of decision making. And that is, can restoring WAC expression improve function in individuals with DESH? That's just a hypothesis at this point. And it's seems like a no-brainer, and it's the hypothesis that most loss of function haploinsufficiencies are going after. But if we can show that it actually does that, well, now we have a case for someone to go after it and treat and invest in treating it, because they know it will make a change. in a human, for our families, for all of us. So answering that question will really help us know where we go next. And if it doesn't express, then we're sort of back at ground zero again, and we need to come up with the next research question and start hypothesizing. So this is what came out of this workshop for me, and I know I've spent the entire meeting sharing it, and so I'll stop.
That was awesome, Caitlin.
I'd agree.
Well
It's
done.
a phenomenal overview and kind of forward step. So thank you.
Yeah, I'm glad we have the AI notes tracking all of that. Thank you, Caitlin. Um, I mean, for that last point, it strikes me then as, it brings it full circle in terms of current situation and clonazepam.
Yeah.
And how does that impact DESH's expression? Excuse me, the
Right.
WAC genes expression. And, you know, in terms of having an investment lens and, you know, share owners who want a return on their investment in investing in things, if there is a um, drug out there which will satisfy, the need of turning that gene back on for whatever sort of still poorly understood mechanism at the moment we have, does that sap some of that initiative to find the long-term solution of an appropriate and specific design genetic therapy? Something I think about when I hear that, and see how that all ties in together. But yeah, did you have any conversation about clonazepam with folks there, and how did that go?
Yeah. So I have a couple of takeaways about drug repurposing and specifically clonazepam. First, to your first point, every we chase every path that will work for a DESH treatment, because what
Yeah.
works for some won't work for others. So if drug repurposing is out there and it's a phenomenal thing, and we need to obviously run with it, but that doesn't stop us from finding the other stuff. And we do this simultaneously. It's not a progression. It's not, like we get after it. The So the very first thing when I mentioned clonazepam, I was sitting at a table with FDA approver. She's a reviewer, actually. She reviews all IND applications for new drugs. And I talked to her about finding therapies, and we really wanted to target behavior. And she said, well, behavior is so, you sounds really like you're going after a symptom therapy. And I was like, no, I don't want to do that. Like, but there was this drug that seemed to like re-express and like fix what we believe is at issue with DESH. And it was clonazepam. And unfortunately, she found it humorous and was said that she goes, you don't want to know what I'm thinking. And I said, well, I'm sure my scientific advisors have told me the same thing. So like lay it on me. And it basically came down to um, anecdotal evidence, and the clonazepam already has an effect on the central nervous system. We know this. So to see some sort of change would seem natural. And the biggest one I think that people keep coming back to, which is why natural history data is so important, is that there is still natural progression. There are still kids that learn to walk and learn to talk. It's just a matter of when. And so it's really hard to predict or to show that a drug is making that happen sooner than would naturally occur. So validating this as something in terms of a behavior phenotype or a cognition phenotype is extremely challenging. But the fact that there's blood testing that upregulates WAC is still an objective measure. So her take on that was that's going to be really tricky to get through any kind of approval. And this is true for a lot of repurposed drugs. So a lot of the folks at this conference have some sort of drug repurposing going on because there are screeners available everywhere for, you could pay someone to run a list for you that could target your syndrome or your specific mutation. And people get their hands on these and then they try to find the drug and they and so it's not a new thing, I guess. It's not unique to us, I should say. But what is interesting is the divide. So this goes back to just getting your kid treated versus following the rules. And on the one hand, you have folks who don't think it is able to be validated and question it. And then the naysayers. And then on the other hand, you have patient advocates who say, you guys are going to take forever to get your act together on any type of study that will get this in the hands of my child. And I'm not going to wait for you. And if it excludes me from a trial, I don't care. There's also a washout period of this drug. So why would I not be able to do that and get on the trial later? And I'm not going to wait around. And screw it. And some of these other groups have actually taken data on their own and just sort of, used it as an observational tool to point them in the direction of other therapies if this is one that won't be validated. But there are a lot of groups that are in our same position with drugs that are thousands and thousands of dollars, that are not on shelves, that cannot be prescribed because they're sitting on the back shelf. And you get into all of these licensing issues of who owns that drug and are they going to then manufacture it because it worked in this one child in this one study, but you can't get it out to everybody. We are so lucky that clonazepam is not like that. Even if you get it without insurance, it's like $15. You can find prescribers, at least in the United States, which
that's the other issue is that with a syndrome like DESSH, where actually the majority of patients are not in the US. There are a lot, but there are a ton of patients in other countries. And in some countries, rules about taking drugs that would otherwise be prescribed for something else are pretty lax. But in many countries, for example, Rob Galloway is trying to get a study started in England. The rules there are extremely strict. They cannot give clonazepam off-label to anyone unless his study gets approved by a pretty tight ethics review. And that's the case in many other countries. So while you can prescribe or get doctors to prescribe clonazepam off-label in the US, it may take a lot more to get it to become available to DESSH patients in other countries or any other drug, that we would find. So it's just another complication as this moves forward. And I think the caution about that you raised about when a drug is introduced is critical as well and is currently an unknown. I remember when we first met as a family with Dr. Shinawi, his view was whatever damage is caused by the lack of a WAC protein by a certain age could not be reversed. So if you developed a treatment and applied it to a child who was 2 versus a child who was 10 versus a child who was 20 versus 30, the accumulation of damage done by the lack of WAC gene could not be undone. You could only prevent further damage going forward. Well, I don't hear them talking that way anymore. That it's 3 years down the line, which seems like almost no time at all. And yet now, they are saying, well, maybe some of the damage can be rolled back. We just don't know.
Yeah. It's kind of like they talked about this as an endpoint in an FDA trial, using just stopping something as an endpoint. If it stops the progression of something, then you can use that to validate that as evidence. If it uh, uh, like seizures, you have a seizure. If they just stop the seizure, like that's enough. It doesn't need to fix or repair anything. That's enough. So if it stopped any type of further stuff that whack does to human later in life, that would be enough to pass as a treatment. But then there's also the very real possibility that it could reverse something or improve something too. Definitely not something structural, like your brain is your brain, your heart is your heart. I don't think introducing a new drug will fix the shape of things, but um, maybe some of those softer skills, those cognitive skills, those behaviors, those executive functioning stuff. I don't know. Maybe there is something. And from a selling point, Alyssa was very smart to tell everyone that we don't think there's an end in development. Like this could really be something that hits everyone.
And, I think something with DESSH we need to keep working toward is for some diseases, even other rare diseases, there is maybe 20 or 30 or 50 years of data on the progression of the illness, like Angelman syndrome or a variety of other diseases. With DESSH, we're dealing with maybe 5 years of data.
Yeah.
We have much less data on the progression, and that's something that as an organization we can continue to help build. If we know more about the natural progression of the disease, we're better able to see where a drug interaction might change that progression. Change
Right.
down progression, upgraded, up progression, or whatever. And it's not just the clinic data. I realize sometimes I beat the drum too hard on the RARE-X data, but you were talking about finding symptoms that we didn't even know about, and yet could be things that could be used to measure a drug's efficacy. One of the things about RARE-X is because as a database, it's almost like a shot in the dark. I'm going to ask you a ton of questions, and it may turn out that 90% of them are not relevant to your disease, but 10% may end up being connected to characteristics of your disease that you didn't even know about, that you didn't know were correlated with your disease, with DESSH in this case, but they become quite apparent. And we've already found some of these in the RARE-X database, and I think we'll find more as things are analyzed that can help.
Yeah, there was another call today actually, that I was on just about the importance of natural history study data. And it's actually a series. There's going to be another workshop, and that really hammers home that what we're doing with the clinic and data collection is right. That's what we need to keep doing and focusing on and collecting. But for a long time, I was also thinking, you hear these timelines. Well, this step in the process of drug development takes 1 to 5 years, and this one takes 10 years. And you talk about people that have like all this plethora of data, which is great for them to go like retrospectively and see, because that's a real asset for them. But I also, my point is, we're not behind because we don't have that other data. And we ought to still collect it, because who knows what happens, down the pike. But we have what we need to get something done already. The mom who got the N of 1 treatment is from Amber Freed, was diagnosed in 2018, and she's been through an FDA approval. There were a couple of other disease groups there who, were just discovered in like 2022. Like, I love to brag, we were just found out in 2015, and they're like, oh, well, we just came around in 2022. And I'm like, and you're here already? It took me how many years to get to like this place, So like there's urgency, and we're really, really, truly, science is moving super, super fast.